Okay, hello, John Dempsey here. This is a presentation I've given over the last year or so at various places such as the Czech Republic, at BTME in Harrogate, GIS Orlando, and this is the latest version which I did on March the 12th at the Golf Course Superintendents Association of New Jersey Education Day. It's called Fertility for Turfgrass Defense, but I'll be looking at a whole range of alternatives to fungicides to control disease in turf grasses. I start usually with a bit of a background check on myself, just to let people know who I am and where I'm coming from. So basically I'm a greenkeeper. I've been working in the turfgrass industry since the 1980s, and I was course manager of the Royal Curragh Golf Course in Ireland, which is Ireland's oldest golf course, since 1993. Of last year I parted ways with them, and I'm now a fully independent researcher into turfgrass disease and physiology. That's my main job, along with going around talking about turfgrass, which is quite nice, as you can imagine. How did I get into turfgrass? Well, it was the 1980s and I didn't actually have a job. My father-in-law was a greenkeeper going back to the 1960s, and he got me the job. He was a friend of mine, David Logan. He's a professor of mitochondrial respiration, I think it is. He reckons that, back in the 1980s, that this was the advice I got. Get into grass, John. They pay you to cut it, and it just keeps growing. And of course, the important piece here was you can make stripes. Well, that wasn't the reason. Obviously, as I said, I needed a job. So I was the second generation of the family to get into greenkeeping. And of course, our son is also a superintendent now at the Slade Valley Golf Club in Ireland. And even our grandchildren, 4 generations, have helped out on the course. So basically what we're saying is that despite going into academia and getting my degree and PhD in plant pathology, any work I've done in education and research was very firmly rooted on the ground. So whatever work I was doing in the laboratory one day was being brought back to the golf course the next. So a very sort of robust research into plant disease and control is what we're looking at. So today's talk is disease suppression with limited fungicides. Basically, we're looking at ways to reduce disease incidence in this time of limited fungicides. And I'm going to use what's called the disease pyramid to target how we as turf managers can actually influence disease factors. So I'll be looking at things like the pathogen infection process and crucially how plants actually defend themselves against infection. Because we need to look at defense priming and enhancement, as this is a vital way to reduce disease incidence. We're going to show some solid scientific data which will support cultural practices and how they can actually influence disease levels. And then we'll move on to fertility for disease management. And putting it all together then towards the end, I'll be showing some field trials which have been carried out, putting all these various factors together, and you can see not only how they can reduce disease, but also some of the modes of suppression. So what exactly is a disease? Well, this is from George Agrostis' book, Plant Pathology, and it's a bit long-winded really. What we're talking about, in my opinion, is a problem caused by a living organism, a pathogen, on amenity turfgrasses, which for us affects the visual and playing qualities of our turfgrass swards. So here's some of the major pathogens we have to deal with in the UK and in Ireland. But the 2 main ones that will cause us most problems are Anthracnose and Microdochium. And we have to look at ways in which to suppress these diseases now with the limited availability of fungicides. So you might ask, why not just control these diseases using fungicides? And a few years ago I was doing these talks and I used to say, well, fungicides can be quite expensive and a lot of us have limited budgets. They can also, of course, inhibit beneficial organisms. And I used to say there's a possibility in the future that these fungicides could actually be limited by legislation. Of course, nowadays we just say there's severe legislative restrictions. So here's an example. These fungicides here, carbendazime, chlorothalonil, iprodion, these were the mainstay of our chemical controls for many years. Propiconazole then, as Banner Max, was also very effective. And of course these nowadays are totally banned. We still have these here on the right, these selective fungicides, these systemic fungicides, and but we don't know for how long. So we have to move on and look at other ways to help us reduce disease levels. So how can we suppress disease with in this time of reduced fungicide inputs? Okay, well, this is the disease triangle, which I'm sure most people have seen over the years. You have a level of disease here in the center, and you have three factors which interact. I actually prefer this here. This is the disease pyramid. And the level of disease incidence is actually influenced by four factors which interact. And we as turf managers have to be able to focus on whatever of these factors we can actually influence to reduce disease pressures. And this is the way we can control disease without fungicides. So first of all, we're going to look at the pathogen section of this pyramid. And like I said, the two major ones we deal with in the UK and in Ireland and most of Northern Europe would be Anthracnose and Microdochium patch. So we'll have a quick look firstly at some of these infection processes. And this is Anthracnose. It's a fungal pathogen, a Ascomycete fungus, Colletotrichum cereale. And in turfgrass we have two forms of it. We have basal rot or foliar blight. Now, basal rot, as you might see from the name, affects most of the lower parts of the plant. It affects the crowns, the stem bases, and the roots. It affects mostly poa annua, and what you will see is the youngest leaf will turn red to black and rot at the base, and it will affect the root system, causing stunting of roots. So, not good. The more common version of Anthracnose is foliar blight, and this is a midsummer problem. It affects mostly shoots. You get discoloration. You get similar to drought stress. You get this yellowing of leaves. Again, it attacks mostly poa annua. Here's a photo of an infected green. So you can see the poa is quite yellow and in bad shape. The bent grass is holding up a lot better. So with Anthracnose, what we're having here basically indicates a problem with the turfgrass. The turfgrass is stressed, is much more inclined to be infected. So this is a fairly basic greenkeeping with Anthracnose. You have to look at ways to reduce stress. But also we have to help the plants to be able to respond to stress in a timely and healthy manner. So you're down to looking at plant health, in other words, to fight off this disease. This is a quick look at the infection cycle of Anthracnose. So you would have spores, the reproductive spores, the canidia, could be splashed onto the leaf area, onto the crowns and the foliage. And then these spores would germinate and infect the plant. So the spore would come onto the leaf, as you can see here in the bottom left. It would then start to grow out, and it would produce what's called aprosoria. And these aprosoria are structures which actually would burst through the outer cuticle of the leaf of the turfgrass and enter the plant. Once the fungus gets into the plant, it travels through the intercellular spaces into the cells and it basically extracts nutrients. So it's in there to eat and drink basically. There's just a few photos here of that particular process. The top left, you can see the reproductive structures, the canidia, and over on the top right you can see one of these canidia which are actually has germinated, formed a germ tube, and you can see the aprosoria, the structure here growing out from it, which will burst through. So that's the way it gets into the plant. As I said, it goes into the plant, extracts nutrients, then exits, and you can see on the bottom photos these black structures here, which are then allowing reproduction of the canidia, which are then spread, and the whole cycle of infection can actually then continue. So we move on now to have a look at Microdochium nivale. The full infection process is what we're going to look at now. So what exactly is Microdochium? Well, like Anthracnose, it's an Ascomycete fungus, and it's known as Fusarium patch or pink snow mold. And in the UK and Ireland, it's the most common and damaging pathogen in turfgrass. It's also the most economically important winter disease of turfgrasses in the northern and alpine regions of Europe, in the US and Canada. Just a few photos here of it. I'm sure you know what it looks like. So, it affects most cool season turf grasses, although Poa annua is particularly susceptible to it. We carried out an extensive study into the infection process of Microdochium, and we had numerous pot samples and various grass species which we looked at in the greenhouses. And also, coming from Ireland, we had no shortage whatsoever of outdoor trial areas and infected greens to use as part of our research. One technique in particular which we found very useful was what's called fluorescence microscopy. And this involved using various stains which attach to the fungal cell walls. And then when viewed using what's called a fluorescence microscope under UV light, these fungal structures become very clear. And you can see here in these photos, the ones, for example, on the left, the same leaf which had been infected, and you can see the top one is under normal light, and then under UV light you can see how the fungal hyphae and that become very clear. Likewise on the right of the screen here, this is the same leaf under varying intensities of UV light. So this process was very useful to track the entire infection process from the source of inoculum right through how the fungus got into the plant, how it traveled through it, and what it did and how it exited and reproduced. So what we found in particular of interest was that we looked at various root zones from previously infected areas, such as here. We got samples and we ran through this staining process. And this is what we found. This is the blue hyphae here. These are the individual strands of Microdochium nivale. And you can see it in amongst the sand particles from a green's root zone. So these particles were there almost year round. In fact, we found them. And then, when environmental conditions were correct for the infection process, the hyphae would grow from the base of the plant up over the sheath and onto the leaves. What we can see here is a creeping bent leaf, and you can see the blue hyphae, the mycelium, covering the leaf. This is just another photo of an infected leaf. You can see the mass of the mycelium. The red is actually the chlorophyll in the plant, which autofluoresces red under the UV light. Again, what we found then was that the Microdochium entered the plant, unlike Anthracnose, which burst its way through the cuticle. Microdochium was more stealthy, and it entered the plant via the respiratory openings, the stomata. And you can see the blue filaments of these hyphae and the circular or semicircular type opening here. And this is where the Microdochium entered and got into the plants. Just another photo here of an infection process, the hyphae entering the plant via the stomata. So once it was in the plant, the individual strands of the fungus, the hyphae, would travel through. Again, it's there to eat and drink, basically, to extract nutrients. Once it had extracted nutrients, it would then re-emerge from the plant again via the stomata, and it would produce numerous, in fact many, of these reproductive structures, the conidia. There's a leaf. This is the same leaf under different types of focus, top and bottom, and you can see the numerous reproductive structures which are being ejected. Again, this is just another photo of these reproductive structures. They're very small. About maximum length of these reproductive structures would be 25 micrometers, so 25 microns, very small indeed. And it's these that are then spread by wind, by water, by feet, and of course by machines. And the whole process then of infection can recycle and continue. So here's the infection process. This is actually relevant to almost any fungal pathogen. The source of the infection, the inoculum, would be in the soil or the thatch layers. And then when conditions, the environmental conditions for whatever pathogen you're looking at, are suitable, the infection process would begin. The mycelium would grow, in the case of Microdochium, from the base of the plant and enter via the stomata. With Anthracnose, aprosoria are formed and would burst through. But again, each various pathogen has its own way of penetrating, but the overall infection process is quite similar for most. They're in there to extract nutrients. Like any other organisms, they just want to eat and drink. And then they reproduce, and the whole cycle of reproduction and infection continues. But for us, who's looking at diseases and at how to control diseases, the crucial part of this cycle is that infected plants will respond with some very complex defense mechanisms. This is just a nice slide of a sort of graphic of showing how a fungal pathogen can get into the plant and extract nutrients. You can see this. You can see where it travels through the stomata here, through the intercellular spaces and into the cells. But again, to emphasize the fact, infected plants respond with some very complex chemical defense mechanisms. And these defense mechanisms what form this interaction in this pyramid of disease between the pathogen, the host plant, and time. Because what we talk about here in time is the time it takes the pathogen to grow and infect the plant, but even more importantly, the time it takes the plant to recognize it's being infected, and the time it takes then to respond with these defenses. So we can influence, if we can slow down the pathogen in some way, if we can give more time to the plant to respond, well then we're going a long way in reducing these levels of disease. This is just a quick overview of some of the plant defense mechanisms. We have both passive or constitutive defense mechanisms which are in place. These are on the left as you're looking at it. These are full-time, they're in place throughout the plant's lifetime. But what we're interested in mostly here is the induced defense mechanisms, which are these defenses which are induced as soon as the plant recognizes being infected. And they can come in two forms. They can be structural. In other words, they're physically strengthening the plant against the infection, or biochemical, where a whole range of defense-related compounds are produced. And it's this area that's of particular interest to us. So here's a sort of an overview, a very brief overview of what's going on when a plant is being infected. These are the cells here. The pathogen would enter by whatever means. The infected cell then would recognize through elicitation that it's been infected. And one of the first things that happens is that the surrounding cells of the infection site would actually be programmed to die. And this is trying to contain or stop the spread of the infection. So we have this short-term rapid defense, and this involves the production of various what's called reactive oxygen species, Key to this is hydrogen peroxide. And this first line of defense is, as I said, is trying to contain or stop the spread of the pathogen. It's called a hypersensitive response, and like I said, the cells surrounding the infection site do what's called programmed cell death. But this is just a short-term rapid response. The more interesting and more complex response involves long-term or systemic acquired resistance. And this involves the production of a whole range of defense compounds. And many of these are what's called phenolic compounds, and they involve production of lignification of cell walls, which is strengthening the cell walls. They produce antimicrobial compounds, such as phytoalexins, and they include signaling compounds, such as salicylic acid. And these all combine to induce a whole plant defense, whereby in future infections, in fact, can be fought off much more rapidly. And these induced defenses, this systemic response that we as turfgrass managers must be trying to enhance or prime the plant with these prior to infection. So the question, of course, is, can these defense mechanisms be stimulated or enhanced? And the answer is, of course, yes. We can look at various things called defense activators. And these are treatments which are applied prior to infection, and they're used to control diseases by priming the expression of these defense mechanisms. Some of them I'm going to mention today would include civitas, chitin or chitinase, salicylic acid, and phosphite. So here's an example of how civitas actually can reduce snow mold severity. What you can see here in the bottom line here is the level of disease in the control plots. Now the rest of these treatments are all various fungicides, but if you look at this one here, this is the effect civitas had on Microdochium levels. And you can see it was very effective at reducing disease levels, purely actually by enhancing these or inducing these defense mechanisms. Again, this is the effect civitas had on Anthracnose. Again, if you look at the top level here, this is the control level of Anthracnose, control plots. Again, you have various fungicides, but the civitas here at the bottom, very effective at reducing disease levels. Again, induction of defense mechanisms, primarily to reduce disease levels. Another defense activator I'm going to mention is chitin. So what exactly is chitin? Well, chitin is a long-term polymer, and it's the main component of fungal cell walls. So it's also a component of the exoskeletons of crustaceans and insects. So this is a source of chitin, and it's been found that when applying products which contain chitin or chitinase, is another form of chitin, these act as elicitors. So you apply it to the plants again prior to infection. The plant recognizes chitin and thinks the fungus is in the plant. So it kickstarts these defense mechanisms. So by applying chitin to uninfected plants, it enhances or primes these defense responses, so that when the plant is infected, the defense mechanisms are already in place. So again, this is one way, another way of reducing disease incidence. Salicylic acid, again, is another defense enhancer or defense primer. Now, when a plant is infected, as I mentioned earlier, we have the first line of defense here in this infected area. So while this short-term response is carrying on, salicylic acid, which is a compound, a phenolic compound, travels through the rest of the plant to the uninfected areas, and it induces or starts off the production of a whole range of defense compounds, so that when the infection, say, spreads, these defense compounds are in place. So again, it acts like an inoculation, And it has been found that applying salicylic acid or forms of salicylic acid to plants prior to infection, that it can actually again reduce disease levels. And over on the left here you can see this is a study which was done at Penn State by Rahman et al. In 2013. They looked at reducing grey leaf spot on perennial ryegrass, again just by applying salicylic acid prior to infection, and they had a very good reduction of this particular fungal disease as well, again just by enhancement of these defense mechanisms. Another one I'm just going to mention is harpin. Again, this is acts as what's called an elicitor. So it's picked up by the plant, recognizes it as a threat, and then it produces the defense compounds. And there's a number of products available, which includes harpin. So again, you have to provide this as in a product, as a program. Because what I have to mention with all these defense activators is that these defense compounds they're not produced out of thin air. So you need to be on a balanced nutritional program, because the plants have to have the resources to produce these extra compounds, because as I said, there's no such thing as a free lunch. So a balanced nutritional program has to be in place for these defense enhancers to be effective. So the last defense activator I want to mention is phosphite. So what exactly is phosphite? Well, it's a form of phosphorus. I'm sure everyone knows phosphorus, a major plant nutrient required for numerous processes within the plant. Now, phosphorus is taken up in plants as phosphate, which is, as you see on the bottom left here, the basic molecule is phosphorus atom with 4 atoms of oxygen around it. And in this way, various enzymes within the plant will lock onto 3 of the oxygen molecules and then incorporate it into whatever process is required. Now, phosphite, as you can see on the right, very similar chemically. It has a phosphorus atom in the center, but only has 3 atoms of oxygen. Now, the thing is that the various enzymes required for metabolic processes will actually lock onto this phosphite molecule. But because there's no extra oxygen molecule or extra oxygen atom available, it cannot be incorporated into the plant as a form of phosphorus nutrition. So it's important to point out that phosphite cannot be metabolized in plants and does not supply a form of phosphorus nutrition. And as a matter of fact, it can be very detrimental to apply phosphite to plants that are in a low phosphorus situation. And just to veer off from disease control for one moment, we did a study as part of our PhD research looking at the effects of phosphite on turfgrass growth. And we had 2 turfgrass species, Lodium perenne, Poa annua, and we grew these in a P-sufficient root zone and a P-deficient root zone and applied 3 treatments. We applied phosphite, and a control. And we looked at the effect these treatments had on shoot, crown and root growth. And this is what we found. This is in a P-deficient root zone. So the turf grasses here were growing in a low phosphorus root zone. And what we noted was that by applying phosphite, which is the green bars here on this, the plants actually behaved or grew worse than as if we applied nothing. And what was going on here was that when all plants are in a deficiency of phosphorus, they will change their mode of growth. The root architecture, for example, will be changed so that it can actually try to extract as much phosphorus as available in the various root zones. Now, by applying phosphite, the plant actually recognized this as phosphorus, and these compensatory changes did not occur. So if you compare the blue bars which were the non-phosphorus treated controls, to the green bars, which were phosphite, you can see by applying actually no phosphorus whatsoever was actually better than applying phosphite. Now we expected that because it has been observed in other plant systems. But here's something that we didn't really expect. This is the effect of phosphite on the growth of the crown, root and leaf of, this is Poa annua, and this Poa annua was growing in a pea-sufficient root zone. And what we found in our study was that by applying phosphite to plants in a pea-sufficient root zone, the crowns, the leaf and root growth was actually enhanced. And we're not really sure the exact means of this occurring, but there could be a possible couple of answers to that, but we're not really sure as of yet. But what's important to note is that phosphite applying to your turfgrass systems, you have to ensure that there is a sufficient amount of available phosphorus either in the soil or in the plant. And you can see in the photo here the effects. You can see the center sample is where we applied phosphate, grew quite well. But control the two outer ones. The one on the right is where we applied phosphite. I compare that to the control on the left where we applied only potassium. Quite a difference. But anyway, getting back to disease control, phosphite, while not very effective as a source of phosphorus nutrition, has been found to be very effective at controlling a whole range of plant pathogens over the last 40 years. Very effective at controlling diseases such as Pythium and Phytophthora, but you have to bear in mind that these are not fungi, they're Oomycetia pathogens. So when I started looking into phosphite and the effects on Microdochium and Turfgrass about 15 years ago, while there was some research on the effect of phosphite on Anthracnose and Turfgrass, there was no published data whatsoever into the effect phosphite had on Microdochium and Turfgrass, which left the door open for us really to be amongst the first to study this problem. So one thing we did was we ran trials for 4 years at the Royal Curragh Golf Club. We applied phosphite alone or in combinations with fungicides when we had them and some biostimulants, and we had 3 turfgrass species in our trials. We had Agrostis canina, which is velvet bent, we had creeping bent and Poa annua. So we applied these treatments and we looked at the effect on the level of Microdochium infection monthly, and this went on for 4 years, 4 seasons, from September right through to March. While we were doing that, of course, we also looked at the effects on turfgrass quality. So this is what we found. This is the mean values of one of the highest peaks we had is January 2011 in Poa annua. But you can see on the right, the blue and green bars are the levels in the control plots. We actually had the blue bar is actually the NP and K equivalent of the phosphite product we were using, except without the phosphite. So the bars on the right show the level of control of disease in the controls. And what we found here on the left, the light blue bars, was the effect phosphite had. We had about a 50% reduction compared to the controls. The fungicide, Microdochium, was very effective. But what we found was the combination of fungicide and phosphite very effective. And you can see that here with the highlighting. Hopefully you can see it with the yellow, which is circling around the C. That was the combination of phosphite and Microdochium. Very effective at controlling Microdochium. This was a similar result we got in December 2010 with velvet bent. Again, on the right, the control level of disease. And again, we found phosphite here on the left, as you look at it, about a 50% reduction compared to the controls. This was the creeping bent in November 2011. lower levels of disease in this, as you can see on the control plots here. But again, phosphite applied on its own, potassium phosphite, very effective at reducing disease. Now, like I said earlier, any of these controls or treatments or cultural practices we're going to look at today will not fully control Microdochium or any disease, but taken in a program with other treatments and other practices, they should combine together to be quite effective at reducing disease incidence. This is the overall picture of the 4 years of the trials we carried out at the Royal Curragh. The 3 grass species on the right, that's the mean values and the control plots. What we found consistently was that by applying potassium phosphite sequentially, the level of disease was roughly halved compared to the controls. We also found, as I mentioned, we looked at the effect of phosphite on turfgrass quality. That's just the overall uniformity, the colour, density of the trial areas. And we found again consistently that applying phosphite improved these qualities overall. This is a better slide here, I think. You can see where any treatment that included phosphite, the level of turfgrass quality over the 4 years was significantly improved compared to the treatments that did not include phosphite. So what we concluded from these trials over 4 years was that sequential applications of phosphite significantly reduced Microdochium nivale incidence, and the addition of phosphite to fungicides significantly enhanced the disease suppression. So you have this synergy of effect of different modes of suppression. And again, we have to say there's quite a significant improvement in turfgrass quality. We actually published those results back in 2012, I believe, and they're available in a peer-reviewed journal here. Just a couple of photos comparing the trial plots from that particular time period. They're all fairly self-explanatory. Poa annua here in January 2011. You can clearly see the difference between the controls and the treated plots. Again, that's the Poa phosphite treated. I think it was January 2011 as well. And a control plot, which was pretty much devastated, really. Again, a creeping bentgrass phosphite treated plot. A velvet bent phosphite treated plot. Just an overall view of some of the trial areas, 2011 again. Nice comparison between phosphite and the control in Poa annua. This is one of the first trials we did in the greenhouse in 2007. So these pots here were growing in the greenhouse. They were the control pots. And then on the very same shelf in the same greenhouse, the phosphite treated pots. So quite a difference. So that was the first trial we did in the greenhouse, and we knew we were having some pretty good results at that stage, which sort of spurred us on to further investigate what was going on. So the trials produced some pretty good results, but we had to delve deeper and see what was the means of suppression. And it could either be 3 means. It could be direct, in other words, the phosphite was actually acting as a fungicide or a fungistat and either killing or slowing the growth of the pathogen. It could be indirect. It could be enhancing or priming these defense compounds I mentioned earlier on, or obviously it could be a combination of both these methods. So what we first looked at was the effect of phosphite on the pathogen itself, Microdochium nivale. And for that we collected our samples and we grew them on PDA, potato dextrose agar, on petri dishes, as you can see here. And then we actually then, that's just a sterilized leaf of an infected plant, surface sterilized on PDA, and the mycelium, the hyphae of the fungus, grew out from the leaf. So we were able to collect it in this way. And then we had a whole range of these petri dishes and PDA. We amended the PDA with a whole range of different concentrations of phosphite, of potassium hydroxide, of combinations of commercial products as well. So the PDA was amended with a range from 0.5 micrograms of phosphite, for example, to 1,000 micrograms per milliliter. And from that we were able to narrow down the range and look at the effect these compounds had on the actual growth of the fungus. As you can see here on the right, you can see the fungus growing on the plates. So this is a pretty good chart here, which we presented in Norway for the European Turfgrass Society conference in 2012. So what we're looking at is the effect of phosphite and phosphate on the mycelial growth in the laboratory of Microdochium. And the bottom line, the flat red line here, shows the effect phosphite had on the growth of Microdochium, and it actually had no effect. The Microdochium grew as per normal. But what we did find then was that increased concentrations of PO3 phosphite in the growth media inhibited and slowed the growth of Microdochium. So after 4 days at 100 micrograms per milliliter of PO3, there was no growth of the Microdochium. This is a good visual showing the effect. So we would have started off on day 1 like this here with a centerpiece of the infected PDA, and by day 4 here in the phosphate amended PDA, it would have grown out almost to the edges. The control was much the same, but after 4 days in the phosphite treated amended PDA, there was no extra growth. Now we carried out further research in this area, and we found that a phosphite didn't actually kill the fungus, but it interfered with the uptake and metabolism of phosphate, and it severely restricted the growth of the fungus. So it slowed down the growth of the fungus. This is a photo of the individual strands of hyphae of Microdochium. The top one is a control, the middle one is in potassium hydroxide, and the bottom one is in phosphate. Now compare that to the ones here on the right, which is the fungus growing in the presence of phosphite. And you can see the individual strands are stunted, and the phosphite is interfering with the uptake, as I said, of phosphate and causing a severe restriction in the growth of it, stunting the growth of the hyphae, slowing it down. So our conclusions from that particular area of study was that phosphite, once it's in the growth media and taken up by the fungus, inhibits mycelial growth and also conidial germination. It disrupts the morphology of the hyphae, and this is causing the fungus to release stress metabolites. So in the plant, once phosphite is in the plant prior to infection, it will slow the growth of the pathogen. It also, by causing the fungus to release these stress metabolites, these would be picked up more rapidly by the plant, again alerting the plant there's an infection occurring and allowing the plant to respond. But while that was one way in which phosphite reduced infection, we thought from the start that the main area of disease suppression actually came from phosphite priming the defenses prior to infection. Now that in vitro work we actually published in 2018, I think it was, again in a peer-reviewed journal which is available in Plant Pathology. So we wanted to see whether phosphite actually could infect or enhance these defense compounds. And again, just a quick overview. This would be an infected area here where the yellow is moving around. So while the infection first occurs, like I said, the plant is trying to contain this infection by the hypersensitive response. But again, the signals are going out to other uninfected parts, and a whole range of defense compounds are being produced. And these compounds are primarily what's called phenolic compounds. They cover a whole range of these defenses. So they're all phenolic compounds. So what we wanted to look at then was whether phenolic compounds were involved in turfgrass defense and whether we could actually enhance them prior to infection. So we did this. What we did was we measured various infected and non-infected plants using various laboratory techniques, and this is what we found. First of all, this would be, I'll put these up. This is Poa annua on the left and creeping bent on the right. Now the blue bars are showing the level of these phenolic compounds in the plant in uninfected plants, control plants which are not infected. The red bars are showing the level of this total phenolic content in Microdochium infected plants. So we can see from this that the enhanced levels of phenolic compounds are equal to the infection. So this is showing that this is a vital response in turfgrass, production of these phenolic compounds. Again, this is what we did in the greenhouse. There's Poa annua on top and the red underneath is creeping bent. And this is levels of these total phenolic compounds in plants which we infected. And at over 11 days you can see there's an increase in these defense compounds. On the left, day 1, compared to say day 6 on the Poa, you can see there's an enhancement of these. So this shows the plants are producing these compounds in response to infection. This is an interesting one here. So we applied phosphite for 6 months sequentially. This is Poa annua we're showing here. We applied phosphite and we applied equivalent rates of phosphate and the untreated controls. And at the end of the 6-month period, we analyzed the tissues and found significantly enhanced levels of these defense compounds in the phosphite treated plants. And this carried through into our greenhouse plants. So this is Poa annua in the greenhouse, which we inoculated with Microdochium. And you can see the red bars are showing the phosphite treated plants, and you can see the difference in the level of these defense compounds compared to the phosphate and the control plants. Much enhanced levels of these defense compounds, much more rapidly produced and in greater amounts. And this actually carried through then to the levels of disease. In the same plants in the greenhouse, the phosphite treated plants had much lower levels of disease compared to the phosphate and the control plants. Okay, this is just an image visual showing an infected leaf. You can see the blue infection hyphae here, but these yellowy green colors here are showing these bursts of these antimicrobial compounds, these phenolic compounds trying to fight off the infection. So what we concluded from that section of the research was that phenolic compounds are a component of defense responses in turfgrass, and that phosphite pretreatment led to enhanced responses in regard to total phenolic compound accumulation. And we also, not only from extracting and measuring the amount of phenolic compounds, we also observed this using fluorescence microscopy. So overall, phosphite pretreatment was enhancing this defense response, leading to lower levels of disease incidence. So that was defense enhancement. So we're going to look at other ways now to reduce disease levels in the absence of fungicides. What else can we influence? Well, we're back here with the disease pyramid. Now we've seen the pathogen, and we've seen the interaction of the pathogen, the host plant, and time, where we can enhance the defense responses. But another crucial area of defense that we can actually influence is so what other factors can we actually influence to reduce disease incidence? So we're back here to this disease pyramid, and there's things like environmental factors. Now all pathogens have various sets of environmental conditions which suit their infection process. So we have to look at areas that, whatever particular pathogen you're dealing with, look at environmental things that you can actually influence. Things like dew removal, for example. Leaf wetness is always a crucial area for reducing disease levels. Things like air movement and light quality. But any areas that you can reduce stress on your turfgrass will help. There's also cultural practices. So we use cultural practices to produce solid, fast and speedy greens. But I'm going to show some scientific data which will also help you to understand that by tweaking these various practices you can actually affect levels of disease incidence also. Then there's also the plant factors here which we can also look at. I mean, some people are looking at ways to totally convert their swards from say for example Poa annua to less susceptible species such as fescue or bent grasses, but you can also look at introducing less susceptible cultivars of whatever species you're dealing with. Balanced nutritional inputs are crucial for turfgrass quality and health, because like ourselves, if we're suffering from malnutrition or if we're grossly overweight, we're much more susceptible to disease. So a balanced nutritional program is also crucial for turfgrass health and disease suppression. But also I'm going to be looking at nutritional IPM, which is basically the use of various nutritional compounds which will have a direct effect on disease levels also. So what about cultural controls? Well, here's something from ICL in the UK. They have a product called DewSmart, which is used to reduce dew on greens. You apply it and it'll have an effect maybe for 3 weeks, depending on conditions. You apply the product and it leads to reduction in dew formation for a number of days, certainly maybe weeks. And this is a research that was carried out at the Sports Turf Research Institute in the UK, looking at the effect of Microdochium on Poa annua, I think it was, certainly on turfgrass. And what you can see here is the blue line here on top is the level of disease in the control plots. Now, by applying this dew suppressive product and reducing leaf wetness, they had a pretty significant effect on Microdochium levels. So again, just by ensuring more leaf dryness, reducing the level of the time of leaf wetness can have a significant effect on disease levels. This is the effect of rolling on disease incidence. This is Dr. Clint Mattox from Oregon State University, who's carried out many years of study now on alternative ways to reduce Microdochium. And one thing he looked at was the effect of rolling on Microdochium incidence. And this is what he found. So the plot here on the left shows levels of disease on a plot that has been rolled weekly over 4 months, and the one on the right shows the non-rolled plot. So rolling is having an effect here on disease levels. This is from Jason Haynes. He's in British Columbia, a nice area for Microdochium also. And this is some study he did looking at the effects of rolling again on Microdochium. And what you can see here on the extreme left is the non-rolled level of disease. By rolling once a day here, you had a reduction in Microdochium, and by rolling twice a day, further reduction. So again, just some evidence showing that rolling can have a direct effect on disease levels. This is from Rutgers University in New Jersey in the US. They looked at the effect of rolling on anthracnose. This is from 2007. They had 2 types of roller, sidewinder and a vibratory roller. And both these rollers found that you would get a reduction in anthracnose, again compared to the controls, as you can see here. This is actually dollar spot, again from Paul Giordano and et al. Here, and they had a look at daily rolling on dollar spot levels, and you can just see from the photos here that again rolling having a direct effect on disease in turfgrass. Now what I think is going on here is the rolling is slightly stressing the turfgrasses prior to infection again, and this stress is actually leading to an induction of defense compounds. Now I haven't any solid data to support that, but that's what my opinion is anyway. This is top dressing, and the effect light regular top dressing can have on Anthracnose in Poa annua. Again, this is from Rutgers University. What you can see here on the top line is the Anthracnose level in the non-top dressed plots, and regular top dressing, the light blue line is twice is more regular than this line here. So regular light top dressing can have again a pretty significant effect on Anthracnose. And you can see in the plots here in this photo, the plots on the right were top dressed regularly, some every 7 days, some every 28 days. Compare them to the ones on the left which received no sand, and you can see quite a difference. So what's going on here? Again, as I said, Anthracnose is very much a stress related disease, and you can see in this photo here that on the left no sand, but what the regular top dressing is doing is protecting the crowns and the lower parts of the plant, thereby leading to less stress, and less stress you have less Anthracnose. This is the effect of mowing height on Anthracnose, again from Rutgers University. So the top line here is the level of Anthracnose at 2.8 millimeter height of cut. By increasing the height from 2.8 to 3.6 millimeter on the bottom line here, you can see pretty good reduction in Anthracnose levels as well. So going from 2.8, increasing the height by 0.8 of a millimeter has a significant effect on Anthracnose levels. Again, you can see the effect here in this photo, 3.6 on the left, 2.8 on the right. So with cultural practices, I've just mentioned a couple of them there. Things like ensuring leaf wetness is reduced, combining lightweight rolling with regular sand top dressing and tweaking the height of cut. You're going to produce pretty good playing surfaces, very good playing surfaces. At the same time, by tweaking your program, you can have a significant effect on disease levels. Okay, we're going to look at plant nutrition as part of this pyramid of disease. And what we want to look at is obviously the nutritional status of your turfgrass swards will have a direct effect on disease incidence. Like I said, plants that are malnourished or overfed are going to be more susceptible to disease usually. So a nutritional balance is the goal. But what we're really going to look at here is the effects various nutrients have directly on disease levels. So here's the thing, like 8 of the essential elements can influence one or more turfgrass diseases. So all these elements or compounds here can have a direct effect on disease in turfgrass. Now one of the most obvious ones is the use of nitrogen and its effects on Anthracnose. So here's something from Rutgers. Now, we're not saying to use these levels of nutritional inputs or nitrogen inputs, but it's certainly highlighting the level of disease that nitrogen can affect. So these high bars here, the levels of disease, are at 73 kgs of N per hectare per year. By increasing the N, you can see a really significant reduction in disease. What we should be looking at during times of high anthracnose pressure is regular spoon feeding of nitrogen. As you can see again from Rutgers is this monthly applications here of N. This is the top line here. But if you can go weekly spoon feeding N into your program, there's a good reduction in anthracnose. So this is a study from Rutgers University looking at the effects of nitrogen on anthracnose. So nitrogen was applied at 4.9 kgs per hectare every 7 or 14 days. Various forms of nitrogen were used: ammonium sulfate, ammonium nitrate, urea, calcium nitrate and potassium nitrate. And what they found was that both N frequency and N source affected disease. So N applied every 7 days reduced disease compared with N applied at the same rate every 14 days. And also they found potassium nitrate provided a greater reduction in disease over the 3-year study compared to, for example, ammonium sulfate. So what we can take from this is that regular spoon feeding, if you can go every 7 days during high anthracnose pressure, will have a pretty good effect on helping to alleviate the problem. So what about iron? Sulfate of iron to reduce disease. Iron has been used for generations, certainly in the UK and Ireland, but does it actually have an effect on microdochium levels? Well, this is a study from Microscopy College in the UK by Jay Usendorp as part of his BSc final year, and what he looked at was the effect of ferrous sulfate on microdochium. So you can see the control level here in the red in the center here. This is the control level of microdochium. Fungicide applied frequently, very effective as you would expect. But ferrous sulfate applied at 10 kgs per hectare on a regular basis during times of high pressure had a pretty significant effect on microdochium. Again, looking at chelated liquid iron to reduce disease. This is from the STRI from ICL in the UK. They carried out a trial in December 2015. Proper scientific design with replicated trial plots on a POA bentgrass sward, high levels of disease risk, and what they found was that applying this line here on top is the control level of microdochium. Applying liquid chelated iron had an effect applied preventatively, a slight effect. But the better effect was achieved by applying this chelated iron curatively at the very first sign of disease. And you can see it had a pretty significant reduction on microdochium. What's going on? Well, some think that the ferrous sulfate is actually acidifying slightly the surface. This is some research I did in the lab. Again, it's similar to the phosphite study where I amended PDA with various concentrations of this chelated iron, neutralized to pH 6.5, and we found that in the presence of the chelated iron at certain levels, the fungus was actually again slowed down, or the growth was inhibited. So the iron was actually affecting the metabolism of the fungus and slowing the growth. So what about potassium as a form of nutrient to affect disease levels? Well, we were always told, potassium was the health element. And again, you can see in this particular photo here that the potassium had a pretty beneficial effect compared to the no potassium. And it's certainly the case with anthracnose that potassium is required to have an effect on disease suppression. Certainly, if you drop below 35 ppm of potassium, your levels of anthracnose will actually be greatly enhanced. So you'd certainly need a sufficient level of potassium in your program or in your plant to ensure that the anthracnose levels are reduced. On the other hand, this is from Dr. Doug Soldat from Wisconsin-Madison. He's been finding lately that lower levels of potassium are actually leading to lower levels of Microdochium. And you can see in this photo here on the right is a plot that has received no potassium for 5 years. The one on the left with much higher levels of Microdochium has been receiving regular inputs of potassium. So if this is a study we'll have to keep a good eye on, because it's very interesting. Manganese and zinc. These are micronutrients, but manganese and zinc have been incorporated in various fungicides for many years, such as mancozeb and dithane. And over the past 2 years, certainly in the UK, a number of products have now been produced which are based on manganese and zinc to incorporate into a program as part of disease suppression. And manganese serves as an activator for various enzymes in these defense pathways. So it would be an important input and important to ensure that there is adequate manganese in your plants to ensure that these defense processes can actually activate correctly. Silica or silicon for disease suppression. Well, this silica is very common in the Earth's crust, but mostly is locked up and not available. How does it have an effect on disease levels? Well, silica can actually have 2 forms of suppression. By applying silica to our turfgrasses, it can build up on the cells and act as a physical barrier. So if you remember with anthracnose, it has to burst through the outer cuticle here by forming aprosoria. So silica can actually help strengthen that. And again, with all pathogens, once they're in the plant, they need to burst through and get into the cells so they can extract nutrients for themselves. So again, silica can help physically strengthen the cell walls and slow down the process of infection. But silica also has a biochemical effect. Again, this is a fungal pathogen, Fusarium oxysporum, which is growing on PDA. And as you can see in the photos here, the presence of silica in the growth media again is acting like we saw with phosphite and slowing the growth and stopping the growth of the particular fungus. So it can have 2 effects. Silica can actually physically strengthen the plant cells, but also has a biochemical effect in reducing the growth of the fungus. And this is just an example from Florida, from Dr. Lawrence Datnoff, looking at the effects of silica on grey leaf spot in a warm season grass. Here on the left is the level of control, quite high. Silica or silicon, as you can see, reduced it. The fungicide chlorothalonil again reduced it. And this is something you see, this combination of different elements and compounds working synergistically to have an even greater effect on disease levels. What about calcium? Well, calcium is crucial, as we all know, for a whole range of metabolic processes in the plant, or many defense processes. As you can see in this sort of diagram here, many defense mechanisms require on the availability of calcium, and calcium can become locked up when immobile in the plant. So ensuring there's elevated levels of calcium freely available is vital for many of these defense processes. Copper is something that's been used actually as a fungicide for centuries, going back to the 1700s in fact, and often as part of a copper-based fungicides up to right through the 20th century. So again, copper in a program or in a product will help to niggle away at disease incidence. Won't fully control disease in turfgrass, but like I said, all these elements and compounds form a jigsaw, and when you put them together, they will have a very significant effect on disease levels. Sulfur. Well, sulfur has been used as a fungicide going back actually to Roman times, so a long time in use in agriculture. Here's a research done in 1975 with Microdochium patch on bentgrass greens. Pretty high levels of sulfur, but they found it did have some control. More recently, in Oregon State, Dr. Glenn Mattox, as part of his research for his PhD, has been looking at the effect of sulfur treatments to Microdochium, and is getting very good and interesting results. So what we can see here is on the right the levels in the control plots, no fertility added, of Microdochium this is. Sulfur applied here on the right, quite effective. Again, this is a polyphosphite here, this is where I'm highlighting at the moment. Again, what we should note is this combination of different compounds together, very effective. So sulfur and the polyphosphite, very effective, working in different manners to reduce disease. This is the same graph, only I wanted to highlight the fact that there's rolling and non-rolling going on. As I mentioned earlier, Dr. Mattox has also looked at the effects of rolling on Microdochium. So all these treatments that were applied here were either rolled or not rolled. And you can see that the rolling had along with the treatments, had a very significant effect at further reducing disease. So this is the recommendations from Oregon State for the use of sulfur to control Microdochium. They found that keeping sulfur levels below 98 kgs per hectare per year and applied from September through to March or April, quite effective. But the combination of sulfur with phosphite was very effective at reducing disease levels. Like I said, it's this combination of different elements and programs can be quite effective. As you can see in this photo here, we have, for example, in the center here, the untreated control. Again, this is from Oregon State University. If you note here at the top left, the civitas, pretty good at controlling. The sulfur here, again, quite effective. And you'll note the combination of these two different elements, even more effective. And it's this synergy of combining various elements and treatments that we're really interested in. So, that brings up the question of, can this nutritional program actually have an effect on turfgrass disease? And what research is there to support this? So down here you can see the various elements and compounds like phosphite, salicylic acid, silica, sulfur, calcium, copper, iron, manganese, zinc, nitrogen and potassium. These are all compounds and elements I mentioned just previously. So what are the trial results? What's the research showing by combining these can we actually have a significant effect on controlling disease? So here's some research again from Oregon State University, and some really interesting results here. For example, have a look first at the number 1 here on the bottom right. This is the control plot, a nice covering of Microdochium in that. Equally of interest to me certainly is this number 7 here, which has been highlighted. This is tebuconazole, fungicide, and you can see some evidence here of Microdochium. All these other examples, these other photos, are various combinations of phosphite, of sulfur, of chelated iron, and pigment. And as you can see, they're all producing pretty good and interesting results. Again, much the same here. This is the control here at number 1. The tebuconazole again with some evidence of disease. But all the rest of these, again, they're various concentrations, different combinations of products and of timing rate applications. Again, mostly phosphite, sulfur, chelated iron and pigments. All very, very effective and looking pretty good. Again, this is just from Oregon State, just a a nice contrast between the control plot here and this combination of phosphite, sulfur, iron and pigment. So here's some of my own particular research projects. Now we saw earlier on the Microdochium and Phosphite trial, which went from 2008 to 2014, where we found that potassium phosphite applied sequentially will have a pretty good effect on reducing Microdochium, reducing it by about 50% compared to controls. So here's two more trials I've done in the last 2 years. One is Anthracnose suppression from 2018, and the other is again with Microdochium from 2018-2019. And these were looking at various combinations of phosphite, of ensuring there's enough calcium, of sulfur, salicylic acid, silica, iron, manganese, zinc and copper. So the program we put together was a combination of all these elements. This is the first one we're going to look at from this Microdochium suppression trial, which ran from September 2018 to March 2019. Again, we had looked at all these various combinations of phosphite, calcium, sulfur, silica, salicylic acid, iron, and we compared these treatments to untreated controls and a fungicide treated plots. So this was the program. This ran from September 2018 to January 2019. So we had an untreated control, we had a polyphosphite here on its own, and then we had this combination of products here, which I'll show what they contained when we get the results. And then we had in strata the fungicide control. So like I said, they ran from September to January, applied every 14 days, and we assess incidence bi-weekly. Then from January to March, we had the same treatment program, but we also included a chelated iron here into the program. So this is what we found. This is the results which we got from October to January 2019. October 2018 to January 2019. So the level of disease in the control plots was here on the right. The polyphosphite here on its own was quite effective, reducing disease from about 50% to just over 20%. The combination or product combinations here had a further reduction, but statistically identical to the phosphite, actually, you should note. The strata was, as you would expect, was pretty good. So a good reduction in disease from that program. Now when we added the chelated iron into the program, again the control is here on the right. The phosphite 30, again pretty good at reducing it, but when we added the chelated iron, we had a further reduction in disease levels, and statistically, if you note, it was actually even greater reduced compared to the phosphite 30 polyphosphite here. Again, the fungicide was effective. So we got a good reduction from just under 40% to just over 10% in our program of nutritional inputs. And I think, looking back now, if we had included manganese and zinc, for example, into the program, we would have got even further reduction. But, it's a step in the right direction, and this can be tweaked and reassessed and get further reductions coming into the future. This is just some photos from that particular trial. The top left here is the control plot in December 2018. This is the phosphite treated plot here. The combination of products which we just looked at is up here on the top right, and you can see the overview there of various treatments. So what was going on here with this particular trial? Well, first of all, the phosphite which we applied on its own was enhancing and priming these defense responses. These other products we put in were ensuring that we had adequate nutrition for that particular time of year. We're also ensuring there was enough calcium available, and phosphite again for defense responses. And we had salicylic acid in the products as well. So this was priming and ensuring for systemic resistance. And then the inclusion of the chelated iron again further decreased disease levels. So this is the Anthracnose trial. This ran during 2018, which was a particularly hot and stressful year for turfgrass and for turfgrass managers, of course. So this trial ran from July to October 2018. We applied treatments every 2 weeks and assessed disease incidence, in this case Anthracnose, weekly. So the treatments were untreated control here. We had a polyphosphite here, and then we had this combination of products here as well. And we had Bannermax as the fungicide. This is the overall range of disease levels we got from this beginning of August through to towards the end of September. And you can see the control levels started off quite low, peaked towards the end of August, and then dropped away. The other treatment plots had a similar curve of infection, only much lower. So this was the peak of our Anthracnose incidence, and this was the end of August 2018. And you can see the level of the Anthracnose here in the control plot on the left here. The phosphite, the polyphosphite here, very effective at reducing disease levels. But this combination of products here, this nutritional program, was very effective, and in fact it was statistically the same as Bannermax, the fungicide. So very effective control of Anthracnose in this particular program. This was the overall levels of disease for that particular trial in 2018. Control here on the left, the polyphosphite, very effective. Again, the nutritional program on a par, significantly the same as the Bannermax fungicide, so a very effective control of Anthracnose there. So what was going on with this particular trial? How were we getting this suppression of disease? Well, first of all, the polyphosphite here was actually priming and enhancing defense responses prior to infection. This particular product here, apart from applying spoon-feeding nitrogen, also included calcium, sulfur, copper, chelated iron, manganese and zinc, and these are all important, as we mentioned earlier on, all important elements for disease control. We also had this product, which again was supplying a balanced nutritional input of NP and K, and also had some polyphosphite in it. This particular product here was a wetting agent, hydration A, which was reducing water stress, ensuring that any irrigation we applied was being optimized, but also it had an element in it which actually helped to solubilize the calcium. The trial areas at the Royal Curragh are very calcareous, so this was ensuring there was again adequate supplies of freely available calcium in the plant. And this final product here again was providing salicylic acid, which was ensuring and stimulating the systemic acquired resistance, this defense response in the plants. So taken in combination, all this program was actually reducing stress, providing adequate and balanced nutrition, but also ensuring that the defense mechanisms in the plant were also fully operational, and we had a very good reduction of Anthracnose with that. So just some take-home points from this particular talk. Certainly as turfgrass managers, we need to look at this disease pyramid, and we need to focus on whatever areas of this pyramid we can actually influence to reduce disease. With Microdochium, we look at all the environmental pressures that are suitable for infection and try to reduce these as best we can, be it reducing leaf wetness or cutting down trees, ensuring greater air movement. Again, I must mention timely use of aeration operations, and for example in top dressing, many turf managers, certainly in the UK, are under pressure to time their aeration programs until late in the year because of various competitions that are on. So if you're going out with heavy top dressing in October, you're creating a really suitable environment for disease infection there. So if you can, try to retime to a more suitable time of year where the environmental conditions are less suited to infection. Again, incorporate rolling if you can into your program. We see that rolling will help to reduce disease. And look at any factors in the microclimate conducive to microdochium development. Whatever small point you can actually concentrate on, it'll all combine to help reduce disease pressures. Again, in the nutritional program, if you can include things like phosphite, like sulfur, salicylic acid, any of the elements that we mentioned today, as part of a balanced, sequentially applied nutritional program, these can be very effective at reducing disease pressure. With anthracnose, pretty much a stress-related problem with turfgrass, so basic greenkeeping 101 here, just try to relieve stress on our plants as much as possible. Cultural management procedures to reduce disease pressure, as we all know, things like adequate aeration, relieve compaction. But we saw that light sequential top dressing, tweaking your heights of cut, will all have an effect on anthracnose. You're reducing stress on the plant, reducing anthracnose levels. Again, you need to spoon feed low levels of N during this time of high anthracnose pressure, and ensure adequate potassium, of course. And your nutrient program, as we saw in the last trial there, can include phosphite, calcium, sulfur, silica, salicylic acid, copper. These will all combine as a jigsaw of disease suppression, and hopefully you can incorporate some of them into the program. You needn't incorporate all, but if you can incorporate some of them, it will have an effect. Like I said, none of these that we looked at today will fully eliminate disease, certainly not on their own. But if you can put together a program of nutritional inputs, cultural practices, and good environmental procedures, you will reduce disease levels. And also, if you still have some fungicides, all this will combine to enhance your fungicide program. So that's it. You can follow me on Twitter if you have any interest, and any questions you can send there, and I'll try and answer them if I can. So hopefully you found this particular presentation okay and interesting, and you can let me know if you did, or let me know any ways to improve it, and I'll try and do my best. Thanks very much for listening, and I'm just going to leave you a vision of my current office in the summer, hopefully. And thanks again for listening. So any questions, you can send them on through Twitter. I'd be more than happy to talk and try to answer them. Stop.